Hello, my name is Dr. Mahmoud Khalil. I'll be teaching you molecular biology, and then subsequent lectures will do gene regulation, and we will understand some of the genetics. So today, we'll talk mainly on DNA and chromosome structure. So we need to describe and identify the components of the DNA and the chromosomes. As well, later on, in a subsequent lecture, we'll see how DNA is formed and how it will be replicated and organized. And we'll describe the replication process in the eukaryotes and the difference between the eukaryotic and the prokaryotic DNA. Then we'll deal with the transcription and the RNA processing, which is concerned with the RNA synthesis. So we'll describe the structure of the RNA, how it's transcribed from the eukaryotic genes, and as well the difference between the eukaryotic and prokaryotic RNA. And as well, one of the main aim of the lectures is to describe the difference between eukaryotic and the prokaryotic transcription. Then we'll talk about the protein synthesis, and we'll describe the steps and the characteristics of this process. And we'll define the genetic codes and the component needs for the translations and the codon recognition by transfer RNA. Then we will deal as a final lecture for the molecular biology part with the post-translational modification. These are a very important modification that is added on top of the protein after being translated, and they explain a lot of the protein functions. We'll describe these, what are these post-translation modification, which is including phosphorylation, glycosylation, hydroxylation, and many more. And this will help as well in the protein folding, and some of the post-translational modification like ubiquitination help in the protein degradation. The main reference of the course will be the molecular cell biology. And this one of the famous, very famous international books that deal with the understanding of the molecular biology. And you can see this is the latest version, 2016. So what are the nucleic acids? Actually, the nucleic acids is linear polymers of nucleotides. And actually it contains the information that determine the amino acid sequence. And so it will determine the structure and function of the protein, which will be translated. So these factories, these cell factories, will select and align the amino acid in the correct order for the protein to be formed. And as well, these nucleic acids catalyze fundamental chemical reaction in cells and regulate the expressions of genes. So actually, nucleic acid function as the brain and the central nervous system of the cell, while protein will carry most of the function that is specified by the nucleic acids. Of course, you can interrupt me any time for questions. Okay, are you hearing me very well?
Yes, we hear you.
Okay.
Yes.
Okay. So let's go first for the DNA, the deoxyribonucleic acid. So it's built, as we said, it's a build of a sequence of nucleotides. It's very stable, extraordinarily stable under almost all terrestrial conditions. It's very important that the error rate of copying DNA throughout our cells is very low. It comes like 2.5 times 10 to the power 8 nucleotide per generation. So this is the rate of error that can be transformed through generation. And the exact replication of DNA, it needs to be replicated exactly to ensure the genetic continuity from generation to generation. For the ribonucleic acid, majorly there are 3 famous types of RNAs, although there is a lot of other minor, although they minor in its popularity, but have another major function. But generally the 3 major types of RNA are the messenger RNA, the transfer RNA, and ribosomal RNA. And actually the messenger RNA the messenger RNA conveys the signal or the instruction from the DNA through the process of transcription in order to be translated in the cytoplasm to form the protein, while the transfer RNA only concerned with carrying the amino acid to recruit the amino acid, the specific amino acid, to the messenger RNA, and by the help of the ribosomal RNA, the process will be aided and form the translational machinery in which the protein is translated.
Doctor?
Yes?
There is a word called terrestrial, you said it before. What
Yes.
do you mean by it?
Terrestrial, I mean like anything on the earth, any environmental factors.
Okay, thank you.
You're welcome. So let's summarize in this slide the 4 major molecular processes that occur inside the cell. If you assume that this is the whole cell and the part in blue is the nucleus, while the part in purple is a nucleolus, you can see that initially, so we just summarize all the 4 major processes. So initially the DNA, which is linear, the first process is a process of transcription, in which in this process, the DNA at a specific regions, which is genes, is transcribed by the help of ribonucleotides, which is nucleotides needed to build the RNA. And you can see this red string is the new RNA that is formed. So the first process, of course, will deal with every single process in details in the coming lectures. So the main aim of the transcription is to produce messenger RNA. This messenger RNA form, it needs to be processed because originally it is named pre-messenger RNA. So it needs some of its part to be removed. It needs to be protected from both ends in order to, you can see now it is transformed from pre-messenger RNA into mature messenger RNA, in which it loses some of its part. We'll understand later what are these parts. And then through the process, the second major molecular process occurring in the cell, which is the RNA processing, this RNA now will have a cap protecting it, and will have a polyadenine tail protecting its terminal part. And now this messenger RNA will be ready to migrate through the nuclear pore to the cytoplasm in order to get attached with the ribosome to form the third molecular process inside the cell, which is translation, in order to form protein. So where ribosomes come from? Actually, the ribosomes start in the nucleolus, and then the large and the small ribosomal subunits migrate to the cytoplasm together with the transfer RNA and the amino acid needed for the process of translation, all combine to form the ribosomal machinery, which bind to the messenger RNA. So now we have seen 3 major processes: the transcription, the RNA processing, the messenger RNA translation, that keep us with the 4th one, which is the process of replication, in which the DNA copies itself through the process of the division, together with the process of division of cells, in order to ensure that each daughter cell will receive an exact copy of the original DNA. So basically, this slide summarizes the 4 molecular processes that occur inside our cells, inside each of our cells, and we will deal in details with each process throughout our lectures. So let's go.
دكتور عندي سؤال.
اتفضل.
يعني ال DNA is composed of genes and non-gene parts، صح؟
ال DNA وده اللي هنقوله كمان شويه. ال DNA في خلينا نقول عليها coding and non-coding. اوكي؟
Yes، الكودينج هو mRNA.
لا خلينا نقول ال DNA لما يبقى coding بيطلع RNA mRNA. اوكي؟ اوكي. وده هنقوله بالتفصيل، احنا لسه بس بناخد مقدمه في الاول. اوكي؟
دكتور كيران
Go ahead.
Doctor, who will R and T?
RNTP اللي هي رايبونيوكليوتايد، هنتكلم عليها بالتفصيل كمان شويه.
Dr. tRNA
Hold on, tRNA, sorry.
When it is synthesized with nucleolus, where is synthesized?
Every RNA is transcribed from the DNA, whatever the type.
Okay, يعني even rRNA is synthesized from the DNA.
All the RNA are transcribed from the DNA. It's only the region that is transcribed from.
Coding region or non-coding?
Yes. So if the gene transcribed a messenger RNA, so that is coding. If the gene transcribed transfer RNA or ribosomal RNA, it will produce an RNA that will not be translated. You got it?
Yes.
Okay.
بس عشان بالهيستولوجي we took إنه ال rRNA is synthesized in the nucleolus.
And you can see from the figure as well here. Okay.
Okay, yes.
نكمل يا شباب.
But please, can you go slowly?
Okay. So let's go for the structure of the DNA. So basically, the DNA and the RNA actually they are the principal molecules that carry genetic information. Of course, the difference is so subtle between the RNA and the DNA polymers. You can see that for the DNA, you have A, G, and C. While T only is found in the DNA and the U only is found in the messenger RNA. So usually the nucleotide, which actually the building blocks of the either the DNA or the RNA, may have 1 or 2 or 3 phosphate groups that esterified by at the 5' OH. So it's kind of linkage. So let's go in some details. You can see these are the structure of one nucleotide. You can see that it is composed of a base, which is either, as we said, adenine, guanine, cytosine, uracil, or thymine. This base is bound to a ribose sugar, which is generally called a pentose sugar because it's a 5' molecule. And then you have a phosphate group. Together, the phosphate is linked to the ribose through the phosphoester bond. And as well, together, the base and the pentose is form a structure called nucleoside. So actually, the nucleotide is nucleoside bound to a phosphate group. This pentose sugar is either ribose, as in case of RNA, or deoxyribose, as in case of DNA. You can see the difference majorly in the existence of the OH at the position 2', which is lacking in the deoxyribonucleotide. So generally now we have 2 types of nucleotides. The only difference between these 2 types of the nucleotides is in the pentose sugar. The pentose sugar of the RNA is having the hydroxyl group on the 2', while the pentose sugar of the DNA, this one is the pentose sugar of RNA, and this one is the pentose sugar of the DNA. Okay, that's why it's called deoxy, because it's lacking the oxygen. So each of these nucleotides will be the building blocks of either DNA or RNA. So here, generally, in our cells, we have purine nucleotides and pyrimidine nucleotides. For purine, it's either adenine or guanine. For pyrimidine, we have either uracil, thymine, or cytosine C. And these are the chemical structure of the nucleic acids. And of course, you don't need to memorize the chemical structure, but you need to understand which nucleotide is a purine and which nucleotide is pyrimidine. For the terminology, As well, this table is for knowledge, but you need to understand that the symbols for a nucleoside monophosphate is AMP, nucleoside diphosphate, ADP, for example, ATP for adenosine triphosphate. And these are some of the terminology of the purines and pyrimidines. As we said, A, G, purines, while pyrimidines are C's, U's, and T's, okay? And these are the terminology through the RNA and the DNA, and this table only for to be familiar with the terminology, and you don't need to memorize this table. So let's go for the structure of the nucleic acid. So originally you can see in the structure, you can see there is a phosphate sugar, a backbone. So there's alternating phosphate sugar. And this sugar phosphate backbone, you can see that each phosphate sugar is bound to a base, which is either adenine or guanine, each phosphate sugar. So all this structure considered to be one nucleotide. All this structure considered to be one nucleotide. That's why all of these will form the building block of the strand of the nucleic acid. If we look closely into the chemical structure, you can see that here is the C, which represents this base, and you can see the sugar, which is here, and you can see the phosphate group, which is here. So you can see here that each nucleotide is bound to the second nucleotide through a phosphodiester bond. It's very important here that the orientation of the DNA starts from the 5' end. You have a 5' end, and you have as well 3' end on the OH. For simplicity, scientists invented a way to easily read the structure and DNA, and you can see in this stick diagram, you can see how the bases are aligned, and you can see there is a starting end 5', and there is a terminal end which is 3'. For extra layer of simplicity, you can see that scientists decided to write DNA in its simplest representation, in which it is usually always right in the direction of 5', 3', in the direction from 5', 3'. So now the order of the nucleotide will be CAG, as you can see.
دكتور بس البيس من موصولة على الثيرد.
What do you mean? مش فاهم سؤالك.
البيس مش موصولة على الثيرد.
انت بتتكلم في الشكل الاخراني؟
لا فوق الستيك دايجرام.
اوكي.
الثيرد هون موصولة على البيس وعلى الفوسفو دي استر بوند.
Okay, this is a stick diagram. It doesn't يعني ليس بالضرورة توضح الاستراكشر بالضبط الكيميكال. It's only written by sticks. فاهم قصدي ولا لا؟
اوكي.
Okay. It's not a chemical structure. It's not a chemical structure. It is a stick diagram for simplicity. فاهمني؟
So can you please repeat هذا الكيميكال directionally of nucleic acid؟
هنقوله في السلايد الجاي ان شاء الله. هنعيده تاني في السلايد اللي جاي. اوكي؟
انا ما لحقت.
اوكي هنقوله تاني. So let's go again for the structure of the DNA. So in 1953, the famous Watson and Crick, these two scientists proposed the DNA exists in a double-strand form. They use X-ray diffraction based on data from the DNA, data from the X-ray diffraction of DNA generated by two other scientists, which is Franklin and Wilkins. So you can see here, together with another scientist which is called Chargaff, he found that the percentage of A nucleotides or bases equals to the percentage of T, and as well the percentage of G is equal to the percentage of C. So you need to know that these scientists early wanted to understand the structure of the chemical structure of the DNA. So today they found the exact structure of the DNA using the high resolution X-ray diffraction studies. And it's the kind of equipment that you apply a crystal for the DNA, and in this machine, due to the diffraction of the X-ray rays, they can understand after analysis the structure of the DNA. So you can see that they found that in order to be uniform structure of double-stranded DNA, the two single strands had to be oriented opposite to each other. So one single strand will start from the 5' end and go all the way to the 3' end, and the other strand will start the opposite, the other way around from the 5' end and goes to the other way. And you can see that C's are bound to G's through 3 hydrogen bonds, while A's are bound to T's by 2 hydrogen bonds. That's why Chargaff have found earlier that the percentage of A usually equal the percentage of T, because both of them bind together, while the percentage of G's equal to the percentage of C's, because both of these bases bind together. So why the DNA is a very stable molecule? Because they found that, or they understood from the structure, the chemical structure, that there are thousands of hydrogen bonds. So you can see each 2 nucleotide here, for example, bound by 3 hydrogen bonds, 2 hydrogen bonds, 3 hydrogen bonds. So all over the DNA, there are thousands of hydrogen bonds, which gives a huge stability for the DNA. And as well, there is something called the hydrophobic and van der Waals interaction between its adjacent stacked adjacent bases. So if you think like these bases are bound together here, and this base is bound together here, so usually you usually see equal distance between each pair of bases. This is a kind of non-chemical kind of van der Waals interaction between these stacked bases. So Watson and the Crick base pair postulated that in order the DNA to be uniform in size and uniform in diameter, for instance, or the width, uniform in the width, a larger purine must bound to a smaller purine, smaller pyrimidine. Again, a larger pyrimidine, 2 ring, must bind to a smaller pyrimidine. So in total, you can see that's why it makes the DNA very uniform, because only addition is 3 rings, okay? And this keeps uniform width for the DNA. حد عنده أسئلة يا شباب في الجزء ده؟
دكتور، هل الاسماء كليات مطلوبه منا ولا بس عشان نعرف انه الاي على التي والتي على السي؟
لا، الاسماء العلماء انا ما بسالش فيها.
ايوه.
اوكي.
دكتور، هلا الوان فان der Waals في الهيدروفوبيك انتراكشنز منتبه
في المكان ده
بيناتهم، يعني
بيناتهم، عشان كده دايما تلاقي المسافات دي ثابته ما بين كل 2 بيز، كل 2 نيوكليوتايد. نكمل. Okay. So
can you repeat the last idea, please?
Which idea?
Please go back to the previous slide. So, the Watson and Crick base pairs.
For Watson and Crick base pair, it's a larger purine, larger binds to smaller pyrimidine. بس.
دكتور، شو البيورين والبيرميدين؟ انا ما قدرت اسمع، ما كنت عندي مشكله.
البيورين اللي هم السي والتي واليو، والبيرميدين والبيورين البيورين اللي هم الاي والجي، الاي والجيز، والبيرميدين اللي هم السي والتي. اوكي.
Okay, thank you.
You're welcome. So let's go closely into the structure of the DNA. So additional layer of information have been arose. So you can see that DNA actually, it's not like this simple form that you can see ladder-like form, but it appears in a right-handed helix. So you can see that DNA is like a double helix. And usually they refer by the name B-form DNA. So it is actually the B form is the normal form in our cells. Okay, so you can see here the two strands, one strand is red and the other strand is blue. And you need to know that as we said in the previous slide, it is anti-parallel. And how did we know from the figure that is anti-parallel? Because this red strand starts with the 5' and ends with the 3', while this blue strand goes the other way around. Okay. So due to the arrangement and the double helix of the DNA, grooves are generated within the structure of the DNA. And actually these grooves, it's like windows through the structure of DNA that you can see the structure of the nucleic acid and the structure of the nucleotide from inside. And these grooves are termed major groove because it's big, minor groove because it's small. Again, due to the twisting and the helix form of the DNA, you can see that grooves are generated. So what is the importance of these grooves? So actually these grooves are sites which provide hydrogen bond. It can bound using a hydrogen bond.
Doctor, first type major groove.
Okay, so you have, sorry again. Okay, in this slide, Watson and Crick have discovered the initial structure of the DNA, in which they use the X-ray diffraction data from two other scientists, which are Franklin and Wilkins. So they decided that, they understood that the DNA had to be formed of a double strand, and this double strand should be anti-parallel. One single strand from the 5' end till the 3' end, and the other strand the other way around. Chargaff add another piece of information in which the amount or the percentage of the in which the percentage of the A's is equal to the percentage of the T's. And as you can see, A always binds with T with 2 hydrogen bonds, while G binds with C with 3 hydrogen bonds. And this huge amount of hydrogen bonds throughout the DNA have given a high stability for the DNA, as well as the van der Waals interaction and the hydrophobic interaction between adjacent bases. So there is a kind of bond between these two adjacent bases, give as well a huge stability for the DNA. For what Watson and Crick as well propose that always a larger purine consists of 2 rings bind to a smaller pyrimidine consists of 1 ring. As you can see here, a larger purine binds always with a smaller pyrimidine, which we say A binds with T, G binds with C. Here, they found as well, in order to be stable, the DNA has to be in a double helix form. And in this double helix form, it is usually right-handed helix. It twisted to the right. The helix is twisted to the right. And they refer to this structure as B form. And normally our cells contain the B form. Due to the twisting of the double helix form, you can see that there is gaps or windows that you can see from to the inside the structure of the nucleotides. These windows, they termed it major groove and minor groove. So what are these windows function for? Actually, these windows will provide hydrogen bond interaction with proteins that need to bind to the DNA. So proteins need to bind to the DNA, will interact and enter the DNA to form a hydrogen bond through this major groove and minor groove. So both of these grooves are actually potential hydrogen donor and acceptor. They can do interaction between proteins like the DNA binding proteins. So the DNA binding proteins can read the sequence of the bases in the duplex DNA by contacting the major, either the major groove or the minor groove, depending on the function of the protein. So here you can see that generally there is the B form, it is the normal form. Normal form exists in almost all in our cells. It's right-handed helix. The one turn in the helix, it's around 10 to 0.5 pair per turn. And you can see that the complete turn takes about 3.4 nanometer. There is another form that exists in rare condition, like low amount of humidity. It's called the A form. And in that case, with low amount of water content or humidity, the B form is transformed to this form, which is called the A form. In this A form, the difference that it is, the pairs are a little bit tilted, معوجة شوية, and the one turn is consist of 11 base pair per turn. So we will see later in some structures like RNA when it's bound to DNA, or RNA which is when it's bound to itself or another RNA, usually take the form of A form that can exist in cells and in vitro as well. So basically this form, it is not a normal form. It only occurs in low humidity. As a comparison between B form and A form, you can see that the overall proportion is different. You can see that both of them are right-handed. It's around 10 base pair per turn, 11 base pair per turn. Then there is a difference in the major groove and the minor groove and the minor groove. The major groove actually in the B form are wide, but in A form is extremely narrow and very deep. And it's vice versa concerning the minor groove. You can see that it is very broad and shallow in the A form, not in the B form. One of the important characteristics of the DNA that it can be bent, and you need to understand that the DNA, it's a linear molecule. So it is important for the DNA to be bended. Why it's important? To provide flexibility for the DNA. And as well, some proteins need to be functional. It need to bind the DNA, of course, as we said before, through the major groove and the minor groove. Once it binds to the DNA, this DNA binding protein, it will make the DNA bend around itself. And this we will see later, and this will facilitate the process of transcription and the process of replication as well. Yes?
What do you mean by the groove is deep?
Deep, عميق.
يعني مثل كأنه مخبى؟
مش مخبى، عميق. Deep. مش مخبى، مش متخبى، عميق.
دكتور سؤالك انت عن DNA بندد او شيء هيك؟
Bended. سؤالك تاني معلش؟
You said about the DNA that it's bended.
Yeah, sometimes the DNA.
Repeat,
okay, sometimes the DNA is bended due to the binding of that protein to the DNA. Okay. we'll see that later in some some of the few slides and next lectures as well. Why the DNA is bended, okay?
Doctor,
Okay,
the
thank you.
condition of low humidity, it does not occur in normal conditions like in our body.
Not major,
Right.
yes, not a major. Okay, income. So,
دكتور
yes,
بس الدي ان اي بايندينج بروتين مهما شو بتعمل
ال DNA binding protein هنتكلم عليها بالتفصيل في المحاضرات الجايه. دي بعض البروتينز عندها القدره ان هي تتحد مع الدي ان اي. اوكي؟
اوكي
Okay. Sometimes it's a very important characteristic of the DNA that it can do. This DNA can be the strand can be separated, and this process of separation of the strand can be reversible. So why it's important for the two strands of the DNA to be separated? Because it's important during the process of replication and during the process of transcription as well. It needs to be separated in order for the process of transcription, in order to build a new messenger RNA over the template. For the replication, it needs to be separated as well in order to be copied to form the new DNA, the daughter DNA. So in the process of, hold on. So the unwinding and the separation of DNA is called denaturation or melting. So basically the two strands can be separated from each other. Either it's normally occurring in our cells, but either it can be induced as well experimentally by increasing the temperature of a DNA in solution. So you can see here that the DNA, the double-stranded native DNA can be denatured, denaturing and leaving the DNA every single strand by itself, and you can induce this process through the use of temperature, okay? So actually the temperature will break the hydrogen bonds, leaving the single strands. And once the single strands are in solution, they usually form random coils. So what's TM? Actually TM, it is the melting temperature that is required to melt DNA in solution, I mean in the lab, in vitro. And this melting temperature of the DNA depend on a number of factors, 4 factors. The first factor, the higher the GC content of the DNA, you need a higher temperature to melt the DNA, because as you know, the GC pair binds with triple hydrogen bond. So if you have a DNA that have many GC pairs, you will have a lot of hydrogen bonds. So you need a high temperature in order to denature or melt the DNA. The ion concentration as well, because the strands, the general charge on the 2 strands of the DNA is a negative charge. So we have this DNA. So we have the DNA actually, and the single strand have a lot of negative charge on the single strand. So once you lower the positive ion in the solution, this negative charge will not be neutralized. And due to the presence of this negative charge, these 2 strands, once the hydrogen bond are broken, they will be they will repel each other. So once the positive ion, which is not neutralizing the negative charge, is low, this means you need a lower TM. The third factor, you can use some agent that already destroy the hydrogen bonds between the nucleotides, like formamide or urea. If you use these 2 compounds in a solution with DNA, it will decrease the melting temperature you need to use to melt the DNA. The 4th factor as well, when you use extremes of pH. At high pH, high extreme pH or low extreme pH, the need for the temperature that you need to melt the DNA will markedly be decreased. Why? Because as we said, we have a strand that is negatively charged. You have a strand that is negatively charged. If you have a high extreme pH, so more negative charge will be added on top of the negative charge, because the solution will be will have a lot of negative charge in the solution. This will lead to more repulsion of the two single strand. So the TM you need, the melting temperature you need, will be low. What if you have a very low extreme pH? That means you have a lot of positive charge in the solution. Because they are a lot of positive charge, the single strand, instead of being covered by negative charge, it will be covered by a total of positive charge. So as well, this will lead to a repulsion of the two strands, because both of them will have a total charge of positive charge. And this as well will need lower temperature of TM in order to denature the DNA. So here you can see,
Doctor,
sorry, okay, go ahead.
are you responsible for this lecture for the quiz on Monday?
ممكن اخلص المحاضرة وبعدين نتكلم الكويز بتاع ماندي ولا مش الكويز بتاع الماندي لو سمحتي؟ ممكن؟
Okay. Doctor, can I ask you a question?
اتفضلي يا فندم.
Concerning the slide before, what do you mean by the low concentration of solution?
Low concentration of solution, low concentration of a positive ion you need.
Can you explain it, please?
When you do, لما تحطي positive ion في السوليوشن كتير، ده مش هيأثر على النيجاتيف تشارج اللي موجودة على الستراند، فهيحصل repulsion force between two strands. فأنت مش محتاجة درجة حرارة عالية عشان تفصلي الستراندز من بعض. فهمتي؟
دكتور
Okay. Thank you, Doctor.
You're welcome.
شو هي الواي اللي بتصير بالترانسليشن والترانسكريبشن اللي هو الاربعه؟
الاربعه دول احنا مش بنتكلم على اللي بيحصل through transcription and translation، احنا الاربعه دول بنتكلم in the test tube. If you want to melt if you want to melt the DNA, okay?
Okay.
Let's monitor how the DNA, they found that DNA have a different absorption to the UV based on the structure of the DNA. So you can see here, when they monitor DNA at wavelengths of the UV 260, they found that the double-stranded DNA absorbs UV less than the single strand. So actually, they can monitor the DNA if it's a double strand or single strand in a solution by increasing the temperature and measuring the UV. If they find the UV, the absorption of the UV at 260 nanometer is very low, that means it is a double strand. If they found it very high, that means the molecule is a single strand. So you can see here, when increasing temperature, the UV shift from a lower value until reaching a higher value. That means temperature is very important in denaturing the DNA, in formation of a double-stranded DNA into single-stranded DNA. Another very good information here, the percentage of the GC, as we said in the first factor that control the TM, depend actually, the TM actually depend on the percentage of the TM. So you can see here, if the percentage of the GC is around 80, so you need around 105 degrees Celsius in order to melt the DNA molecule. While if the percentage is 40, you need only around 86, 87 degrees Celsius to melt the DNA. And this line chart describes how the GC content in the DNA is important to determine in solution, in the lab, in the test tube, how, what the temperature needs to separate the DNA. To simplify more this example, you can see here, if you have a DNA that consists of these nucleotides, if you count the GC, you will find 1, 2, 3, 4, 5. This is the GC. Of course, I'm talking about one strand. So the percentage of the GC here will be 5 over the whole, will be 5 over 10 times 100, it will be around 50%. If you look from the graph, you will see that 50% will correspond around 90 degrees centigrade, 90 degrees Celsius. So you can understand now how the GC content is important in determining the melting temperature of the DNA.
دكتور
Yes?
يعني ال UV light كمان هو مثل factor في melting of the DNA.
لا هو مش فاكتور للميلتنج للدي ان ايه. اليو في لايت او الابزوربشن بتاع اليو في لايت احنا بنستعمله عشان نعرف الدي ان ايه ده حصل له ميلتنج ولا ما حصلوش ميلتنج. لان هو لو ما حصلوش ميلتنج الدي ان ايه هو بياخد وهو دبل ستراند الفاليو بتاع اليو في بتاعه قليل قوي زي ما انت شايفه هنا. طبعا انا بعمل الكلام ده جوه تيست تيوب فانا مش شايف في التيست تيوب ده الدي ان ايه حصل له ميلتنج ولا لا. فالناس استخدموا اليو في لايت عشان يقدروا يعرفوا اذا كان هو حصل له ميلتنج ولا لا. فبالتالي لما الفاليو بتاعت اليو في تطلع قليله يبقى انا بقدر افهم ان الدي ان ايه ما حصلوش سيباريشن. لكن مع ان انا بزود درجه الحراره اليو في هتلاحظوا ان اليو في القرايه بتاعته بتعلى لغايه ما بتوصل للماكسيمم. معناها بفهم كده ايه ان الدي ان ايه حصل له سيباريشن. فهمتي؟
شكرا.
دكتور
ايوه
يعني فينا نقول كلمه الجي سي بيرز انكريز الميلتنج تمبريتشر انكريز.
ايوه ما ده اللي انا قايله في السلايد اللي فاتت بالظبط. تمام.
دكتور يعني على طول بيستعملوا 260 nm it's a constant.
ده الويف لينس اللي بنستخدمه عند القياس النيوكليك اسيدز concentration. اوكي.
دكتور less single stranded but doctor UV lights.
السنجل ستراند بيابزورب a lot of UV light. اوكي.
شو السبب لانه بده UV light؟
عشان السيرفيس اريا بتاعته اكبر. اوكي.
دكتور هيدي القاعده اللي تحت محفظه او كيف؟
دي مش قاعده حضرتك ده اكزامبل.
تمام.
ازاي تحسب؟ البرسنتج بتاخد الجي والسي divided by the total number of nucleotide times 100 عشان توصل للبرسنتج of GC. ومن الكيرف بتقدر تعرف من البرسنتج بتقدر تعرف how much TM انت عايز. اوكي. نكمل. So once in a test tube you have done, if you need to do a melting, sometimes you need to do a renaturation, actually to bring back the DNA into the double strand. If you need to do this renaturation, you need only what you need is to reverse the condition. Actually, you need to lower the temperature. If you increase the ion concentration, you need to lower the ion concentration. If you used extreme pH, you need to neutralize the pH. Plus, all these three factors will lead to the reassociation of the two complementary strands to form a perfect double helix. Plus, the DNA strand that is not related in sequence, if you have two single strands that are not related in sequence, they will exist in the solution as a random coils. They will never pair with each other. And of course, you will not renature. So this process, of course, you ask yourself, why we need to do this denaturation and renaturation in the lab? This process can be used to study if two DNA samples are related. And as well, they can isolate a specific DNA molecule in a mixture containing numerous different DNA sequence. By the help of denaturation and renaturation, you can compare if two different DNA samples are closely related or not, or you can isolate a DNA sample from different DNA samples. So let's talk about.
دكتور عن أي sequence كان عم تحكي كنت هون؟
أنا بقول
DNA that are not related in sequence.
أنا بقول in general دي فوائد عمليه الديناتوريشن اند ريناتوريشن. اوكي، لكن ما كنتش بحكي على سيكونس معين.
اوكي، بس هذا النوت ما كتير فهمته بعد نمبر 3
لأنه
دي ان اي ستراند.
هذا الانبول.
الاند بول دلوقتي انا لو عملت ديناتوريشن لدي ان ايه، خلاص عندي مثلا خدت الدي ان ايه من انسان، وخدت دي ان ايه من مونكي مثلا، وعملت للاثنين ديناتوريشن، هيبقى عندي كام سنجل ستراند في السولوشن؟ 4. 4. لو انا عملت بقى ريناتوريشن، هل ينفع السنجل ستراند بتاع المونكي يمسك في السنجل ستراند بتاع الانسان؟
لا.
طب هيعمل ايه لو ما لقاش حاجه يمسكها؟ السنجل ستراند هيفضل موجود في السولوشن از راندوم از سنجل ستراند، بس ما بيفضلش بقى ماشي في السولوشن ده سنجل ستراند، بيبقى عامل زي راندوم كويلز لافف على نفسه يعني.
اوكي،
فهمت؟ and will never renature لان هو اصلا مش كومبلمنتري. فهمت؟
حتى لو لقى كومبلمنتري
لا طبعا، لا طبعا لو لقى كومبلمنتري هيمسك فيه.
اوكي تمام.
لكن اللي مكتوب بولد ده معناها ايه؟ ان هو لو ما لقاش الكومبلمنتري.
اوكي
فهمت؟
دكتور في فكرة البي اتش.
البي اتش انا لو استخدمت اكستريم اوف بي اتش تو سيبريت ذا دي ان ايه، بعمل ديناتوريشن اللي في دي ان ايه، لو استخدمت اكستريم بي اتش بمنتهى البساطه، انا لو عملت النيوتراليزيشن هيرجع هيحصل ريناتوريشن للدي ان ايه. فهمتي؟
طيب اوكي.
One of the important behavior of the DNA that happened during the process of transcription and translation is the torsional stress. What is torsional stress? Actually, you need to understand that many bacterial or viral DNA are double-stranded circular molecules. So these circular DNA molecules also exist in mitochondria and in chloroplasts of the plants. So during the replication or the transcription of circular or our DNA, the DNA twists on itself like a twisted rubber band, forming a structure called supercoils. And these supercoils induce a lot of stress on the remaining portion of the molecule because the ends of the strand are not free. يعني عشان نفهم القصة دي تعالوا نشوف كده اهو. ادي دي ان ايه عندي، ده بيبقى دي ان ايه، شايفين يا شباب؟ اوكي؟ الدي ان ايه الدي ان ايه اتفتح من هنا اهو عشان يحصل له مثلا عمليه الترانسكريبشن ويطلع منه الماسنجر ار ان اي. لما حصل له، احنا بنتكلم نورمال انسايد ذا سيل، when it's opened من هير، اكيد نتيجه للاوبننج وعمليه الميلتنج بتاع الدي ان ايه هنا هيحصل كعبله في المنطقه دي، صح كده ولا لا؟
يس ولا صح.
الكعبله دي اسمها ايه بقى؟ اسمها تورشنال ستريس. اسمها ايه؟ لان انا كل ما بفتح الدي ان ايه كده انا بزق بريشر على بقيه الدي ان ايه، فبيبدا ده يتكعبل، صح كده ولا لا؟
طب ليه بيصير بقى؟ صح.
صح. طب ايه الحل دلوقتي؟ يبقى لازم يبقى في ميكانيزم في جسمنا تو ريليف التورشنال ستريس ده، لان once the torsional stress is not relieved, if the DNA is replicating, the replication will not move through this complex part. صح كده ولا لا؟ So we need to relieve. لازم احل مشكله التورشنال ستريس، لازم احل مشكله الدي ان ايه لما بيتكعبل، لما اجي افتح الدي ان ايه بيحصل كعبله في بقيه الدي ان ايه، فلازم احل المشكله دي. طب نحلها ازاي؟ قال لحضرتك ان there are two enzymes exist in our cells. One is called topoisomerase 1 and the other is called topoisomerase 2. Once there is a torsional stress in the DNA molecule, this topoisomerase 1 can bind at random sites and relieve this torsion stress by breaking only one strand, which is called nick, so to relieve the torsional stress. Once the torsional stress is relieved, the same enzyme will bind again the two broken ends. So what's the difference between topoisomerase 2 and 1? Actually, topoisomerase 2 do the same like topoisomerase 1, but instead of nicking one strand, it make breaks in both strands of double-stranded DNA to relieve the torsional stress and then religate them. عشان افهم القصه دي يا جماعه، عارفين انتوا لما يبقى عندكم رباط عارفين شو ليسز، عارفين الشو ليسز ولا لا؟ رباط الشوز خلاص لو اتعقد منك انت محتاج تفكه، صح كده ولا لا؟ مش ساعات لو اتعقد زياده قوي فبتضطر تقطع منه حته بالمقص عشان تفكه ولا لا؟
بلا صح.
طيب وبعد ما تفكه وانت عايز الرباط ده تاني بتعمل ايه؟ بتروح رابطه تاني في بعضه، صح ولا لا؟
بلا.
ده هي دي فكره التوبو ايزوميريز 1 و 2. بس الفرق ما بين التوبو ايزوميريز 1 و 2 ايه هو؟ ان التوبو ايزوميريز 1 بيكات اونلي وان سنجل ستراند، وبعد ما يفك التورشنال ستريس بيربطه تاني هو. اما التوبو ايزوميريز 2 بيكات الدبل ستراند، وبعد كده يروح جاي رابط الدي ان اي بعد ما يخلص. عشان نفهم زياده نبص المنظر ده. ده المنظر بتاع السوبر كويلد دي سيركلر دي ان اي، وده المنظر بتاع الريلاكس دي ان اي. So you can see here there is one single strand that have been cut. So that's why it's called nicked. خلاص؟ ولو بصينا للانيميتد فيجر اللي تحت هنا هنلاحظ ان التوبو ايزوميريز 1 اللي هو اللون الاورنج ده، توبو ايزوميريز 1 هيقعد على الدي ان اي، هيقطع الستراند اللي هو لونه بلو، هيعدي منه الستراند الاحمر، بيفك الكعبله ويربط تاني الستراند. فهمتوا الفانكشن اوف توبو ايزوميريزز ولا لا؟
Yes.
Okay. Let's talk about the organization of the DNA. Actually, the sequence of the DNA and the entire genome have been known for a lot of organisms at the beginning of the 21st century. By now, we already know a lot of other organisms, including the roundworm, the C. elegans, the fruit fly, Drosophila, mice, and the human as well. They found that the DNA actually revealed a large portion of genome that do not code messenger RNA. They are called non-coding DNA. So these non-coding DNA actually a huge percentage is around 98.5% of the human DNA. And only 1.5% which is actually encode functional RNAs. What about the rest? Originally they called the non-coding DNA, they call it junk DNA. And this non-coding DNA, they found the non-coding DNA, which is around 98.5%, actually consists of introns, repetitive DNA, regulatory sequence. And for the repetitive DNA, they are either telomeres, VNTR, variable number of tandem repeats, and transposons. All these are considered to be non-coding DNA. So they are considered not to produce a functional RNA. You need to know that they have other functions. They are not as they already named it before junk DNA. So actually the lens...
They are called dark DNA.
Sorry?
They are also called dark DNA, those 98%, 97.5%.
Dark?
Yes, dark DNA. They have some mysterious functions and other known functions, but almost above half of the functions are not known yet.
Still, yeah, a lot of functions are not known as well. I agree with that. But still, they are not, the major nomenclature for this region is non-coding DNA, because basically they are non-coding. They don't produce proteins. Okay?
دكتور هل من الهيتروكروماتين صح؟
جزء منهم الهيتروكروماتين but not for sure يعني actually 1.5% only that codes proteins. Okay?
Yes.
Okay, here the lens, we need to organize this very long molecule, which is around 2 meters, to put inside a nucleus, which is diameter is around 10 micrometer. So now we'll talk about the organization of the DNA. So in eukaryotes, the DNA is bound to histones. It is a specific eukaryotic protein, and together the DNA and histones is called the chromatin. And you can visualize this chromatin when you see the individual chromosome during mitosis. So basically, what are the functions of the chromatin? Actually, chromatin is very important for packaging the DNA to fit into smaller volume inside the cell, which is actually in the nucleus. And as well, maintain integrity of DNA, because DNA by itself it's a molecule that can be torn in the process of mitosis. So it needs to be protected by chromatin in order to maintain its integrity. The third function, later on we understand that the chromatin provide amine or access to facilitate the transcription, the replication, the repair, and recombination of a DNA molecule. So actually, these are the three major functions of the chromatin. So let's see here, this is a single linear DNA molecule. We'll talk now about organization of the DNA to fit inside the nucleus. So you have a DNA molecule. The first level of organization that these DNA are wrapped around nucleosomes to form, wrapped around the histones to form a structure called the nucleosome. And all of these together will form structure somewhat higher level of complexity, which is called beads on a string. Soon after that, the beads on a string are twisted around each other, like you see here, form another layer of complexity is called the 30 nanometer fiber. And actually 30 nanometer fiber, it is the width of this structure. It is the width of this structure, the 30 nanometer fiber. Soon after that, this 30 nanometer fiber again is looped around itself to form loops of 30 nanometer fiber, which soon form higher order of chromatin folding and will be packed inside the nucleus. And you can see here how many layers of complexity. You can see 1, 2, 3, 4. 4 layers of complexity in order to fit the 2 meter DNA into the nucleus, which is around 10 micrometer. So basically, histones are 5 types: H1 to A to B3 and H4. If you need to remember that histones are the proteins, eukaryotic protein, that DNA used to wrap around it. These histones are.
السلايد اللي قبل.
ايوه اتفضل. اتفضل.
هي الدي ان اي 2 متر بس يعني هي بس في ستراند وحده دي ان اي بقلب النوكليوس ولا عدة دي ان اي ستراندز بتساوي بقلب النوكليوس؟
It's one molecule DNA طوله 2 متر.
اوكي يعني بس وحده دي ان اي موليكيول بقلب كل نوكليوس.
Yes, one DNA molecule in each nucleus of the cell. Okay.
Okay.
So you have histones. These histones, of course, is a positively charged. This positive charge, of course, will bind strong with the negative charge of the single-stranded DNA. So you can see here the structure of the nucleosome. Actually, you can see that the DNA here, you can see that the DNA is wrapped around this core histone. And this core histones actually is an octamer, 1, 2, 3, 4, 5, 6, 7, 8. The octamer actually is 2 copies of each of these histones. You can see that the wrapped DNA around the histones, and the size is around 147 base pair is wrapped around the histones, while the linker DNA, which is the region of the DNA which is connecting between each nucleosome, is this linker is around from 10 to 90 base pair. So you can see here, if you look closely here, this region is called linker DNA. This region as well is called linker. It is the region connecting the nucleosomes. So DNA of the nucleosomes is less susceptible to nucleases, which are enzymes which cut the DNA. Of course, obviously, because this DNA which bound to histone is protected, while the linker DNA the nucleases can, if it got access to the DNA, it can cut the DNA at the linker DNA. There is as well H1 histone, and H1 histone actually, it's a kind of locking down the entry of the DNA and the exit in order to be firm. يبقى ال H1 ده أكني زي بيقفل على النيوكليوسوم عشان يحافظ على الاستراكشر بتاع الدي ان اي اللي ملفوف حوالين النيوكليوسوم. You can see the yellow part is the H1.
دكتور بس ما فهمتش هو الكور.
الكور هو كمية الهيستونز اللي جوه دي كلها. اوكي؟
فيه البيس بير.
نعم.
فيه بيس بير.
بيس بير صح. Actually, the histones are conserved. What's meaning of conserved? The meaning of conserved that the sequence of amino acids of histones among the organisms are very look alike. It's the same. For example, the histone 3, because if you remember, we have histone 1, histone 2A, histone 2B, histone 3, histone 4. Histone 3 of the sea urchin, it's a kind of organism, and the calf sims differ only by one amino acid, while histone 3 of the garden bee differ from the calf thymus only by 4 amino acids. So that's mean that the histones among organisms to some extent are very similar in structure, very similar in sequence of amino acid, except for minor differences, and that's why we say it is conserved. So this similarity of sequence will imply or indicate that the way of the 3D conformation is the same. There are minor histone variant exist as well. There is a kind of histone H2X, which can replace its histone H2A in the core histones in some fraction of the nucleosome in which this type of histone 2X, it helps in the process of chromosome repair process when the DNA are double-stranded breaks. يعني اللي عايزين نقوله ايه؟ الطبيعي ان بيبقى موجود في الكور هيستونز اللي هم الثمانيه، الكور هيستونز الثمانيه اللي ملفوف عليهم الدي ان اي بيبقى موجود فيهم H2A. لكن ال H2A ده بيحصل له ريبليسمنت ب H2X another histone لو الدي ان اي حصل له دبل ستراندد بريكس. والعلماء لقوا ان ال H2X كان بارتيسيبيت في عمليه الريبيرنج، بيساعد في عمليه الريبيرنج. Two other histones like اللي هو اسمها SNPA and H3.3 can replace histone 3 as well, but for different functions. Because histone SNPA can participate in the process of binding to the spindle microtubules during mitosis, while histone 3.3 replaces H3 in transcribed regions of DNA. فاللي عايزين نقوله هنا ايه؟ ان الدي ان اي لافف حوالين الكور هيستونز اللي احنا قلناهم الاربع انواع دول، لكن في بعض الاماكن في الدي ان اي هنلاقي اكزامبلز الثلاث انواع من البروتينز دول سواء كان H2X او SNPA او H3.3 بيعملوا replacing the types of histones in order to participate in other specific function. Let's see the structure of the 30 nanometer fiber. We have said that is formed of wrapping of the beads on a string. So you can see there is a one-start helix structure. It's called the solenoid model. And this solenoid model, you can see that the beads on a string is arranged in spiral form like this. And this is the solenoid model, and sometimes it's called the one-start helix. And this is the widely accepted criteria of the conformation of the 30 nanometer fiber of the DNA. While the second structure, which is strongly supported, it's called two-start helix, and it's called as well the zigzag model. So you can see the difference here. The spiral is one start, so every round is like one step. But here you can see the spiral have skipped two turns. So that's why it's called the two-start helix model. And this is the only difference between the one-start helix proposed model and the two-start helix proposed model, or the solenoid model and the zigzag model. And both of them are theoretical propose of the 3D structure of the 30 nanometer fiber. So yes.
دكتور بس هيدا السلايد مفتوحة كثير.
في السلايد ده بيورينا شكل ال 30. الناس بيقولوا شكل ال 30 نانومتر فايبر ده شكلها في 3D ستراكشر عامل ازاي. اوكي؟ في نظريه بتقول ان هو شكلها 1 ستارت هيليكس، في نظريه بتقول شكلها 2 ستارت هيليكس. الفرق ما بين الاثنين ليه السهوله عشان تبقى
2 point of view.
سوري؟
في 2 point of views يعني هنا.
ايوه 2 points of views to understand the 3D structure of 30 nanometer fiber. فهمت؟
وهي اساس قالوا انه 1 ستار او 2 ستار.
نعم؟
اللي قال 1 ستار على اساس قال 1 ستار، وعلى اللي قال 2 ستار على اساس قال 2 ستار كده.
على اساس حسابات وسايزز وكل الكلام ده كله، قال ان 1 او على اساس الاكس راي ديفراكشن، قال ان ال1 ستارت بيبقى شايف المنظر عامل ازاي؟ هيليكس كده بالمنظر ده، لكن هنا الهيليكس اوسع شويه، شايفينها؟ اوكي؟
دكتور بس فيك تقول لنا اذا هيدي اللكتشر داخله بالكورس تبع التنان؟
ما اعرفش والله. اسالوا الكوردينيتور بتاع الكورس لو سمحت.
طب نحن خلصنا هلا شيء؟
فاضل 5 دقائق.
هي
بس شو الفرق بيناتهم؟
نعم؟
تعيد شو الفرق بيناتهم؟
الفرق the only difference in the way of arrangement. In the way, these are proposed models. The only difference is the way of arrangement of the nucleosome. شايفه السبايرال هنا ماشي ازاي؟ شايفاه؟ هنا السبايرال تحس ان هو اوسع شويه، وما بين كل تيرن وتيرن there is 2 layers، صح؟ هنا ما بين كل تيرن وتيرن هنا ما فيش layers خالص، صح؟ تمام؟ اوكي. ممكن نوقف الاسئله شويه بس اخلص الفاضلي بتاع 2 أو 3 سلايدز بالظبط بس. اوكي؟ بعد ما اخلصهم نسال الاسئله اللي انتوا عايزينها. اوكي يا شباب؟ اوكي. So actually during interphase the chromatin is dispersed throughout much of the nucleus, but during cell division the cell need to fold and compact the DNA to produce the visible chromosome. So actually chromosome exists in 2 forms. You can see here this is actually electron microscope of the structure of beads on a string, while this electron microscope showed the structure of 30 nanometer fiber. So what's basically the difference between both of them? This is a higher complexity in the structure of the chromatin. And scientists understood that when the DNA exists on the beads on a string, that's the mean the genes are transcribed. The genes are actively transcribed, while here the genes are not being transcribed because it is in compact state. So here we can see the metaphase chromosomes, how it is condensed in order to decrease in length and generate this condensed chromosome, which can be observed only during mitosis. Only during metaphase of the mitosis. So each chromosome consists of 1 linear DNA molecule, as you can see. And there is a lot of protein. One of the important protein is the condensin. And this condensin actually is a protein complex, consists of a structural maintenance of chromosome. This is the name of this complex of protein. And it contributes to the process of condensation of this chromosome to be shorter version in order to facilitate the process of mitosis. And this is formed under the expense of ATP hydrolysis. So during the condensation, there are some events. So you can see initially, these are the 30 nanometer fiber that we have been talking on last 2 slides. Further condensation leading to 100 to 130 nanometer fiber called the chromonema. Soon after that, the middle prophase chromatid is formed, which is around 250 nanometer in thickness. And then we'll form the metaphase chromatid. And you can see that its thickness now is 750 nanometer. So you understand now how the DNA try to compact itself with the protein to form with the chromatin to form the chromosome, which is a rigid structure that facilitates the process of mitosis. So ultimately, the
لازم نحفظ اللانثس ولا لا؟
نعم.
اللانثس لازم نحفظهم إنه نكون عارفين.
كل حاجة ممكن لازم تعرفيها. Ultimately, the two associated daughter chromosomes, of course, you understand that there are two associated daughter chromosomes, they can be generated by DNA replication, and they condense into two chromatids, and soon they bind together by the centromere to form the popular chromosome that we all know. So basically, chromosomes number differ among organisms. So you can see one chromosome only in Escherichia coli. You can see 6 chromosomes only in the fruit fly, Drosophila melanogaster. And you can see in Homo sapiens, there are 24 chromosomes, single chromosome, 24 chromosomes. Obviously, in humans, each cell contains actually 23 pairs of chromosomes. 22 is called autosomes, and 1 sex chromosomes that differ from male and female. The last two slides, we are talking about that scientists have managed to use a banding technique, and they can stain chromosomes inside the nucleus of these cells by fluorescence dye. And they found that each chromosomes actually occupy a certain region inside the nucleus. It's not dispersed all over the nucleus. And then they managed to stain each type of chromosome, and for sure they found that each chromosome has a specific territory inside the nucleus. It's not moving around. And this gave a lot of understanding of how the organization of the chromosomal regions inside the nucleus. So these, as we said, these banding technique have distinguished parts of the chromosomes, and they understood that these dye can selectively stain regions of chromosomes. And these banding techniques as well have helped the scientists to understand if the chromosome have abnormality or not. So this chromosomal painting is very powerful method for detecting if the chromosome there is chromosomal anomalies, and when comparing these chromosomes with different species. So you can see here, you can see that the chromosome have been karyotyped, and you can see each chromosome have a different color. And of course, you can see this, if you consider this is a normal pattern karyotype of the chromosome, when you compare it with another individual, you can see if there is deletion or addition for any kind of chromosome. The perfect example here you can see in a distinct case, which is a chronic myelogenous leukemia, in which there is a translocation between chromosome 22 and chromosome 9. So scientists stain chromosome 9, stain chromosome 22. In individuals that have this kind of condition, they will have 2 chromosomes in which 2 regions have been translocated between chromosome 9 and 22. And this actually is a real fluorescent image. And you can see the normal chromosome 22 is stained in green, while the normal chromosome 9 is stained in orange fluorescence dye. Once there is a translocation happening between chromosome 9 and 22, you can see that DER9, what's meaning of DER9? That's meaning it is chromosome 9 have a part from chromosome 22, because chromosome 22 actually is green. That's why it's appearing green here. And you can see here the Philadelphia chromosome, which is DER22. That's mean this chromosome, which is 22, have taken small part of chromosome 9. So actually, the existence of these translocated abnormal chromosome that arise due to the translocation is very indicative of this, which is called Philadelphia chromosome, is very indicative of this type of condition of leukemia, of cancer, which is called chronic myelogenic leukemia. And from this slide, you can understand how it's important, the process of staining of the chromosome to identify any chromosomal anomalies or abnormalities. And this concludes our lecture for today. And of course, the reference we have said in the initial slides, from that reference, you'll see chapter 2, chapter 5, chapter 8, and this actually the number of pages that is required from each lecture. And thank you, and waiting for your questions. شباب اللي عنده سؤال ممكن يتفضل.
الستادي ده ريفرنس ولا الباوربوينت؟
الاثنين.
يعني سايد ذم ولا هو ريدنج بس؟
الاثنين. الماتيريال كلها عليك.
دكتور شو اسم الريفرنس؟
الريفرنس هرجع لكم تاني، هو الفيرست سلايد. Hold on.
يعني بندرس الباوربوينت ونقرا البيجز؟
الاثنين عليك.
Doctor, today we covered chapter 2.
لا أنا ما خدتش كل chapter، أنا خدت بيج 45 46 بس.
Okay.
وكذلك من chapter 5 page 167 until page 174. وكذلك من chapter 8 page 301 302 309 327 ل 329 وهكذا. فهمتوا؟
دكتور سؤال،
ده الريفرنس. لحظة ده الريفرنس بتاعنا يا جماعة اللي احنا بنستخدمه، هتلاقوه في المكتبة عندكم. اتفضل سؤالك.
دكتور يعني نحن بس سؤال، عندنا 23 pairs of chromosomes، يعني نحن عندنا 23 molecules of DNA.
سؤالك ممتاز. انت في حاجة ملخبطك في السايز اوف DNA، صح كده ولا لا؟
انا كنت افكر